Thank you. So I'm a neuroscientist and a neural engineer at UCSF. My laboratory does both basic investigations into the way that we move and the way that we learn to move. But we also study some clinically relevant applications, and that's what I'm going to tell you about today. So just briefly before I get started, I'll say that the work that I'm going to tell you about is the work of many, many people who are currently in my lab and have previously been in my lab. And of course, we couldn't do the work without support, mostly from federal funding sources. So what I want to tell you about today is what we refer to as brain-machine interfaces. And when I say that, what I mean is devices that allow us to communicate directly with the brain. And there are lots of things that you could do when you're communicating with the brain. For example, you could read information out of the brain. Like, for example, you could read out movements that I want to make, or speech that I intend to make, or even memories. And those could be used to help patients for whom these functions are impaired. You might also want to write information into the brain. For example, if you have a blind subject, you could restore vision or hearing or something like that. And then further along, I mean, these are things that we're working on now. And then a little bit further along, you could imagine if you can read out and you can write in, and there were parts that were broken, you could sort of jump over them. You could read out the things that use those parts and write into the things that those parts project to. And so this is the sort of thing that people are thinking about quite a bit these days. And I'm going to tell you about one particular aspect of that, which is brain-machine interfaces for the restoration of motor function. And there are a number of patient populations for whom this would be helpful. For example, upper limb amputees, spinal cord injury, and other forms of paralysis, such as ALS or Lou Gehrig's disease, MS, or other populations. So there's a fairly large population of people for whom the ability to move has been lost, and if there were some way to regain that ability, it would be fantastic, and even if it meant, for example, getting a brain implant. So what would that look like? Well, what is it that you can get out of having a device like this? Well, you have to interact with something in the external world, right? That's the point, is that you want your brain now to control something, not through the normal means, through your spinal cord, your muscles, your limbs, your hands, instead to interact directly with some external device. And for example, there are really beautiful new bionic limbs that have been developed mostly through the support of DARPA, the Defense Advanced Research Projects Administration. For example, this one was developed at Johns Hopkins, and a similar one at Dean Kamen's group, DECA Research. And this is a real photo of this limb that can do pretty amazing things. It looks pretty dexterous. You can also interface with wheelchairs and computer interfaces. For example, here's a picture of Stephen Hawking, who speaks through a synthetic voice by communicating with the computer. Imagine if that could just be read directly out of his brain, and he could speak or he could move as he wants. Now, these are external devices. You could also imagine reanimating your own body, say, for example, through exoskeletal devices, little robots that sort of clamp to the outside of your body and let you move, or even through what we call functional electrical stimulation. And that's where you directly stimulate either the spinal cord or the muscles to allow your own body to move. And these are also things that are currently under investigation. So if there were a way to get information out of the brain in a really precise and accurate way, these are the sorts of things that you could get for that. And that seems like those would be really good things. So how is it that you communicate with the brain? How do you get information out? And most of the devices that are out there these days work by communicating with the part of the brain that's called the primary motor cortex. So this is a side view of a human brain. And the human's sort of looking this way like I am right now. So this is the front of the brain, this is the back of the brain, visual cortex, temporal lobes, frontal lobes, parietal cortex, and this bit here, this strip is the motor strip. So it's the thing that sends projections down the spinal cord and drives activation of the neurons that eventually drive the muscles. So this is the thing that if you stimulate, you get muscle movement. Okay, and because this is sort of the last stop for outgoing motor activity in the brain, it seems like kind of a natural place to record activity to figure out what it is that a person wants to do. If you can decode what's going on here during natural movement, for example, then you can imagine using that decode to control a robotic limb to mimic what would have been the natural movement. That's the idea. So how do you read out information? Well, you have to get something in there to read information out. Neurons communicate with each other via chemical synapses, but those chemical synapses are activated by electrical activity. So neurons basically are electrical beasts. They send what we call action potentials, which are sort of waves of electrical activity down their axons, and that causes information to transmit to the next neuron down the way. And you can read out that electrical activity by putting electrodes, which are just basically little wires, inside the brain, and then hooking those up to an amplifier, and you can get electrical activity out. And this little device here is 100 electrodes, and this is the device that's currently approved for use in humans for these sorts of applications. Not, I should say, for clinical use. These are all preclinical trials, and so they're not. You can't go to your doctor now and ask for this, but there are ongoing clinical trials in small numbers of human patients. And these are, so here's an example of a human with one of these implanted. These are also the devices that we use in my laboratory, as I'll show you in a bit. So how do they work? What happens when you have one of these? So what I'm going to show you now is, this is the raw electrical trace. It's been filtered a little bit, but this is a filtered electrical trace that you would get if you put one of these in a monkey brain. And actually, let me just back up. This is a human brain, this is a monkey brain, but the structure is very similar. So this is what you get if you stick one of these in a monkey brain. And these are we're going to trigger, every time this thing goes high, we're going to trigger, we're going to plot what we see. Can you hear that? So this is a little sound, that little buzzing sound. Those are action potentials. Those are the spikes of a neuron. So we're listening to a single neuron in the brain talking to its downstream neighbors, which is super cool. And it's really cool. When you're a physiologist, you sit and you listen to this, it's really soothing to the soul to hear this. And it's the way that neurons communicate. And you can imagine that if we could read this out, we could decode the information from those neurons and use that to control something. Now, the science of how that works and the algorithms and the information content and how it works, all deeply interesting. I don't have time to tell you about it today. If you guys want to know about it, I can tell you more later. But this is the kind of thing that you can get out of that. So this is from Lee Hochberg and John Donahue and colleagues at Brown University and Mass General in Boston. And this is sort of pretty much state of the art. So here's a woman that has a brainstem stroke, so she can't move, she's tetraplegic. And she has one of these devices implanted on her head. And she's using the readout of that to control this robotic limb. And here she's going to take a drink from this thing, which is something that she hasn't been able to do, to give herself a drink in a long time. And you can imagine that for someone like this, to be able to give herself a drink would be highly satisfying. And indeed, look at that smile, highly satisfying. So this is truly remarkable, right? Here's a woman who can't move on her own. She's moving a robotic limb to give herself a drink. Fantastic. But it's not quite natural behavior, right? This was very slow. It took a long time. I was able to tell you a lot while that video was playing. And in fact, you might have noticed some jumps in the video. That's because I actually cut out some stuff. So it actually takes even longer than that. So this is remarkable and wonderful, but it's not really at the level of natural movement. It's not what you and I would do if we were to walk over there and pour ourselves a cup of water and drink from it. So what's wrong? What's missing? What do we need to get to that level where you can go out and be Luke Skywalker or whatever, and move around with this limb that really is natural? So you might think, well, the problem is, this 100 electrode thing, that looks pretty crude. We've got millions of electrodes in our brain. That's the problem. We're not recording from enough electrodes. And that's true. The neural interfaces that we have today are really impoverished compared to what we want to do. And in fact, in my lab, we're working on some pretty kooky ideas for how to improve those devices, which again, I can tell you about later if you're interested. But 100 neurons is still enough to get a pretty high fidelity readout. So maybe that's the problem. There's probably more. Maybe the algorithms that people have developed aren't good enough yet, though people have done a lot of work and they're pretty sophisticated. So I think it's true that with better neural interfaces and better algorithms, we probably can do better. But I don't think that we're going to get to natural levels of control with just more engineering. There's something that's missing. And what is it that's missing? So I'm going to show you a video and hopefully, good, okay. So here's a guy, and he's trying to sit down. He's just trying to get off his wheelchair and sit on the ground. And if you're looking at him, something's not right, right? I think you'd all agree. So what do you think is wrong with him? Anybody want to guess?
I'd like to guess, but it's probably not what you probably.
Well, go ahead and guess.
Okay, I'm going to guess that he might be severely dehydrated.
Severely dehydrated. Well, he's in a wheelchair, so he's got more long-term problems than that.
Cerebral palsy.
Cerebral palsy. Good guess. What's that? Myositis.
Partial paralysis.
Partial paralysis. Okay, so most of you say things that look like motor disorders, right? Clearly this guy's having trouble moving, right? Guess what? His motor system's fine. There's nothing wrong with his motor system. What this guy's problem is his sensory system. He can't feel his body. That's his problem. Motorically he's fine, but he can't feel his body. By the way, it's a great documentary, and it's actually about something different. It's about a guy who sort of overcame this problem, and you can find it on YouTube, so I recommend it. But in any case, so what the point of this video is that this guy can't feel his own body, and that movement is perception, and perception is movement. If you can't feel your own body, you're gonna have an awful hard time moving. Now, what he's actually lacking is two things. There's something called somatosensation, that's the general term. And that includes tactile things like the ability to touch and feel what things are, texture and the warmth of touching your partner or something like that. And then there's what we call proprioception, which is the ability to know where your body is in space. For example, I can do this. And the reason I can do that is because I know where my body parts are because I can feel them. And that's a term that we mean proprioception. So somatosensation is broken into tactile and proprioceptive feedback from the periphery. So just to give you an idea, that was a guy, that problem was mostly due to lack of proprioception. And here's an example of what happens if you don't have tactile feedback. So this is a video, hopefully we'll play, if not I'll go start it, of a woman who is going to, she's been asked to simply light a match. And this comes from Roland Johansson. This is a really beautiful video, or maybe not. If this one doesn't light, if this one doesn't start, we'll just try this one. All right, well, you know what, if that's the one we missed, that's okay. The point here is that she's asked to simply try to strike a match, just pick up with one hand a match and strike it. And that's a very easy thing to do. She could do it in 7 seconds normally. And then what Roland Johansson did is he put some lidocaine in her fingertips. That's the stuff you get in the dentist to make sure you can't feel your teeth, right? So she can't feel her fingertips. And it turns out that if you do that, it's very, very difficult to manipulate any small thing. And what the video shows is it takes her a long time to pick up the match and light it. And if you want, I can show you that later on my laptop. It's quite an exciting demo, actually. It's really neat. Okay, so the point of this is that somatosensory feedback is absolutely critical for effective movement. And therefore, we think that a naturalistic performance with a brain-machine interface will require somatosensory feedback. It'll give you better control, as I've told you. And the other thing it'll do is, if you're putting on some device, it may feel foreign, it may feel unfamiliar, but if you can feel it, the thinking is, it'll feel like it's part of you. You'll have better ownership of that thing, and then you'll want to use it. So that's the idea. All right, so how can we get artificial feedback into the brain? That's what I want to address next. Well, it turns out, this idea is kind of out there already. The idea of using electrical microstimulation to stimulate neurons to create sensations. That's out there. Many of you probably know someone who has a device. There are tens, hundreds of thousands of people who have these devices, I should know the number, a cochlear implant. So what a cochlear implant does, this is for deaf individuals. What you do is you put, there's a little microphone out here, but basically the core of it is, it feeds into an electrode which wraps around the cochlea, which is the organ, the peripheral receptor organ for sound. And it stimulates the cochlea. It's stimulating the neurons in the cochlea, which causes you to perceive sound. How does this work? Why does it work? It works because the cochlea, here's a blow-up of it, has a very special organization. Some of you may know this, which is that as the sound comes in, which is just waves of air, right, air fluid by the time it gets inside the inner ear, the frequency of oscillations in here is sort of broken down by the structure of the cochlea. So that cells, for example, here will respond to high tones, 600 hertz. To low tones, 200 hertz, 600 hertz. And as you get further along the cochlea, you get higher and you get lower and lower tones. At the beginning it's high, and at the end it's low. So the cochlea has what we call a topographic organization, which is that neurons in the cochlea respond in a way that breaks out by frequency. There's a map of frequency of tones on the cochlear surface. Again, topographic organization. And the reason why topography is important is because it allows you to do what we call biomimetic stimulation, which is to say that it allows you to mimic natural sensory patterns. If you can take a sound, music or voice, and break it up into sort of the intensity at different frequency bands, which is like what your equalizer does, on your computer, then you can play back different amounts of frequency depending on those, okay? So this allows you to decompose the sound into frequency. So topographic organization is critical to give you biomimetic stimulation. Will that work for somatosensory feedback? Well, sort of. So if you look at the somatosensory cortex, which happens to be right next to motor cortex, this is the part of the brain that if you tickle your body or move your limb, these cells will respond. There's this gross topographic organization, which we call the homunculus. So for example, parts of the body that you use a lot, like your face and your hands, have a large number of cells, a large number of areas involved. There's this organization. So that suggests that there's a topography, which means that you can create natural patterns of activation, so you can create percepts. Does it work? Yeah, it kind of does. So here's an experiment from Simon Ben Smaya's lab in Chicago, where they put some of these same arrays that I showed you before into the brain of a monkey in somatosensory cortex. And they touch different parts of the monkey's hand. And say when they're touching here, for example, these neurons light up. These are the ones that would be over here in somatosensory cortex. And when they touch here, these neurons light up over here. So there's a map on the brain that corresponds to the hand. So that's great. That means that you could stimulate now, use electrical stimulation. Here, you'll feel touch here. Here, you'll feel touch here, and so on. And it turns out this works. And you can get monkeys to feel different fingertips and different intensities, and you can have them compare those to natural sensation, and it really works. You can start getting this sense of tactile feedback that feels quite realistic. So this is very exciting, right? So this means that maybe it's not that hard to do the task that we're trying to do. But there's a problem. For proprioception, for knowing where your body is, remember that was the problem that the guy in the wheelchair had. There isn't a good topography. So nearby neurons have very different behaviors. And because of that, because there's no topography, we can't create these biomimetic patterns. And so that raises a real challenge. How do you give feedback? How do you tell somebody where their body is that they need in order to get precise control if you don't have that topography? And it turns out this is true of most of the brain. Now there are many areas, visual, auditory, somatosensory areas, that have these nice topographic maps. But when you get away from what we call early sensory areas to sort of the higher order parts of the brain, there are many, many parts where there just isn't good topography. How do you interact with these? Well, that's not what I have to do. That's what I have to do. So our approach is, instead of focusing on creating natural patterns of activity, let's use the way the brain naturally learns. Let's focus on learning. Now it turns out the circuits for sensation and movement are highly adaptive, and this is something we've been studying for a long time in my lab. And in particular, the brain is very good at combining information from multiple modalities. So vision and audition, as I'm speaking, you can look at my lips, you can hear me, and effortlessly your brain's combining that information. What the brain is really good at is picking out patterns, finding things that move together in the world, and using that information. And we know that, we've studied that in the lab, we've modeled it in my lab, and we're going to try to harness that. It may not be obvious how, but here's the trick. This is work that was done by Maria Daudelat, a graduate student in my lab, and Joey O'Doherty, who's now going to take the experiment that I'm telling you about now and move it really forward to real brain-machine interfaces. So the idea is, what if we write something into the brain that's entirely novel, entirely non-biomimetic, but it changes moment by moment with vision, and we know the brain's good at picking out these sorts of correlations. Could it use it to learn to interpret this novel signal? So here's the experiment. We had the monkeys do kind of a weird task. They were asked to zero out this flow. So they're sitting here at the starting point, and these dots are moving in this direction. So now they know they need to move in that direction. And they get close, but not quite, so the dots are slow. And it's that sort of weird dot feedback scheme. And I can explain why we did that later. But they get good at this. This is pretty easy. And we have them do this for a while. And what the dots are telling them at every moment, the difference between where they are now and where they need to be. So it's sort of like telling them the error of where they are now and where they need to be.
How are they moving the big dot?
So they're just moving their limb. They're just sitting at a table and moving. And that little big dot is following their fingertip around. Thank you. All right, so they're just sitting there doing this with their hand. And then what we're going to do is they get good at this. And then what we're going to do is we're going to stimulate the somatosensory cortex. And we're going to do it with a pattern. I'm not going to tell you about the details of it, but it's a complex pattern that involves 8 electrodes, beep, 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 buzzing along, stimulating local neurons nearby. And it's something that they've never experienced before. It should feel very strange. But moment by moment, it correlates with what they see. So here's a typical thing during training. The monkeys are doing the same kinds of movements I showed you before. So they're supposed to be reaching over here, and the dots are telling them what to do. But then there's this weird pattern being stimulated. Each of these little green markers is a single stimulus that drives a bunch of neurons nearby one electrode. And there are 8 of these electrodes, and they're coded by direction, but it doesn't really matter. The point is that they're feeling some crazy pattern. And they're doing this over and over and over again. Now, this thing at first just feels weird, and actually it impairs their performance. So they do better when this isn't there than when, and there's just vision, than when they're both there. But very quickly, after about a day, they learn to ignore this, and they do it with vision. And that's the way it could have stayed. It could have ignored this forever. But I've already told you that the brain loves patterns. It loves to put things together. And so it can't help but learn what this means, because it co-varies moment by moment with this. Until, after a few weeks, after several weeks, the monkeys can now make reaching movements without any vision at all. They're sitting in a dark room, but they know what to do because they can feel where that target is by the stimulation pattern that we're putting into their somatosensory cortex. And not only that, they're combining these two signals in a very natural way, in this kind of optimal and adaptive way that we know people do with normal sensory cues. Okay, so what do you make of all this? So we call this a learning-based approach to artificial somatosensory feedback, or sensory feedback generally. And the idea is that you learn through these structures of correlations of senses in the world. Again, what we're very good at is picking out patterns, picking out correlations. We can't help but do that. And the signal is integrated in a very natural way. It works together with vision to reduce errors, which is what our normal ability to combine cues does. And just to mention that these experiments were motivated by computational models, which is a very important component of what we do in my lab in trying to think about how to harness the brain's power. This is a new benchmark for writing task-relevant information into the brain, multiple electrodes, 2-dimensional signal, distance, direction, and real-time feedback. So it's a pretty exciting result that we're very pleased about. And the other thing about it is that it's kind of it's one of the first attempts to try to think about how you work with brain areas. How do you communicate with brain areas that don't have a simple, natural, easy to understand topography, like the cochlea, or like the tactile parts of somatosensory cortex. And we think that learning how to do that is something that will generalize quite well to a lot of problems going forward in thinking about interfacing and communicating and repairing the brain.